From the process of southern hybridization, which studies DNA, the process of northern hybridization was developed, which studies RNA. RNA samples from a cell are isolated and run through a gel. These RNA samples are usually isolated from their poly-A tails, indicating that these are mRNAs. The RNAs are then absorbed into a membrane, and then a probe studies the presence of specific RNAs. There are disadvantages to this procedure in that RNA is easily denatured by RNases both in the cell and environmental contaminants, and RNA can form secondary structures which can interfere with its travel through the gel. Also, there are no restriction endonucleases for RNA, although in some procedures the RNA can be converted to DNA by reverse transcription. While the study of DNA allows the identification of a gene, the study of RNA allows one to study the expression of the gene. There are many applications of northern hybridization, which studies the presence of specific RNA molecules. First, while it is known that a cell may possess a specific gene, it is not known whether it expresses that gene. And so a northern blot can identify is an mRNA being expressed from this gene by using the DNA sequence of the gene as a probe. Interestingly, the expression of genes in a complex organism such as humans can vary from cell type to cell type. So some genes are expressed in liver cells, but not bone cells. Genes can vary in their expression during development. So some genes may be expressed as a fetus, but not as an adult. Some genes vary their expression after specific events, such as an infection. So in response to an infection, a certain gene is expressed. Certain genes may be upregulated or downregulated in cancer. So when comparing a cancer cell to a normal cell, one might see a change in expression pattern. Also, northern blots allow one to study alternate transcripts, so that from one gene, the exons can be spliced together in different ways, and so that the protein resulting from a gene in a liver cell might be different from that produced by a bone cell using the same gene, because different exons are spliced together. And this could be studied using northern blots because the RNA band which binds to a DNA probe might be of varying sizes.